So good evening, everybody. My name is Elchanan Bornstein, and I'm an associate professor at the Department of Genome Sciences and the organizer of this public talk series. And before I introduce our speaker, I just want to make two quick announcements. First of all, as always, there will be some refreshments after the talk outside. Our speaker was kind enough to agree to stay for some time after the talk. So if you didn't get time to ask your really important question immediately after the talk in the Q&A part, do feel free to talk to him and ask him all the questions that you might want to know. Second, I know that often we have people that want to talk to somebody from the Genome Sciences department, asking a question about the talk or asking a question about the department in general, and often they just don't know who else in the audience is from this department. So I want to ask everybody that the faculty, a student, a postdoc in this department to raise their hand. Okay. Everybody else, find the person that is closest to you that's raising their hand. This is the person that you can bug after the talk with all those important questions. And really do that, and we're happy to provide all the information that you might have. With that said, this is my absolute honor to welcome our speaker tonight, Dr. Matt Kaeberlein. And in his talk and in his research, Matt was able to do really an extraordinary thing, which is to combine two things. One thing that most of us, at least everybody that is above 20, absolutely fear, and another thing that most of us, including the one under 20, absolutely love, because he's going to talk about aging in dogs. And specifically, he'll tell us about his work in trying to identify the molecular mechanisms that are involved in aging, his work on trying to slow aging and aging-related diseases in dogs, and potentially also how we can apply some of those understandings and translational application to humans in order to better understand aging and to treat aging in humans. Before I'll invite Matt to the podium, I just want to tell you a bit about his background. And so in preparing for this talk, I searched for things that I didn't know about Matt, and I found this beautiful article in Science in 2002. It was already a big deal back then. The two things that I learned is that back in high school he liked basketball and he wanted to be a heavy metal rock star. You still do? He ended up being a very different kind of star, as you can realize. He went on to do an undergrad degree in mathematics and biochemistry in Western Washington University, a PhD in biology at MIT, and a postdoc at this very department before joining the Department of Pathology, where he is now a professor. He has a list of awards that is way too long for me to go through, literally, and I'm not going to do that. I'll just say that in addition to his main appointment in pathology, he's also the director of the Dog Aging Project, the co-director of the University of Washington Center of Excellence in Basic Biology of Aging, the founding director of the Healthy Aging and Longevity Research Institute, and the president of the American Aging Association. So it was not clear until now he really knows what he talks about when he's talking about aging. And so with that said, I'll invite Dr. Kaeberlein to the podium, and I hope that you'll help me to welcome him. Thank you.
Okay, thank you, Elchanan, and thank you for the invitation. It's a pleasure to be here. I did not realize when I agreed to give a talk on dogs that they were going to put me on a leash for this talk, so I will try not to escape. So I've been interested in the biology of aging really since I was a graduate student and before I had so many gray hairs of my own. And really interested in trying to understand, what is it about the aging process, or what are the factors that influence the way that animals and people grow old? And one of the things that's always fascinated me about aging is that when we look at individuals, we don't all age exactly the same way. And I think we can all think of examples of this. So for example, you might not, just looking at these two people, think that Patrick Stewart is actually older than Keith Richards. And one of the reasons why I like this particular comparison is it makes the point that aging is not only about genetics. So genetics, or genome, plays a role in the aging process, but it's also about our environment. And so I like this particular comparison. So this is actually a picture of Patrick Stewart when the Seahawks won the Super Bowl against the Broncos, woohoo, doing a one-arm push-up in his Russell Wilson shirt. And for those of you who know something about Keith Richards, you will probably appreciate that some of his lifestyle choices may have influenced his apparent aging process as well. So So it's about both genetics and environment, like any complex trait. And the goal of my research, and pretty much everybody in the geroscience field, the basic biology of aging, is to understand, well, what are these genetic and environmental factors that cause some people or some animals to age well, stay healthy, live a long time, and others not to. So that's kind of the big picture. And one of the ways to think about why this might be important is to think about disease. So all of us are familiar with thinking about health and disease. And so these are some of the diseases and functional declines that are the major causes of disability, morbidity, and mortality in developed countries. And we're used to thinking about risk factors. So if you go to the doctor, he or she is going to probably tell you that your risk of developing these diseases has something to do with your genetics, your family history, but it also has a lot to do with your environment, how much you eat, how much alcohol you consume, whether you smoke, how much you exercise. All of those things are true, but what sometimes gets lost in this is that none of those things are really the biggest risk factors for any of these diseases. In fact, the single greatest risk factor for all of these things is how old you are. So it's really aging that drives your risk of developing any and all of these different declines in function and diseases. And I think sometimes it's actually useful to look at the numbers, to actually look at the data for this. So these are some of the major risk factors that people talk about for heart disease. So if you have high blood pressure, if you smoke, if you have high cholesterol, if you have diabetes, your risk of developing heart disease goes up between 50% to 80%. So about a twofold increase in risk. If we compare this to your increase in risk just from a 20-year period, going from being 45 years old to 65 years old, so that data is still here, right? And this is what happens with aging. So it really is the greatest risk factor for heart disease. We can look at cancer. So this is all cancers. Not every cancer is age-associated, but most cancers are. And again, if you use tobacco, if you consume alcohol, if you have a poor diet, if you have certain kinds of infection, your risk goes up. But if we go from 45 to 65, again, your risk is much greater. And probably the disease that many of us think about a lot as an aging disease, Alzheimer's disease. Again, we know that some things increase your risk of Alzheimer's disease. So ladies, I'm sorry, but you're at about a twofold risk compared to us men for developing Alzheimer's disease. This is the greatest genetic risk factor for Alzheimer's disease. If you have the wrong allele of ApoE, you have about a fivefold increased risk of Alzheimer's disease. Here's what happens when you get old. Again, just 20 years, 45 to 65. So aging really drives all of these diseases, or at least it creates a permissive biological state that increases your risk for developing these diseases. So the big picture then of the research that I'm working on and many others in this field are working on is that if we can understand what this is, what is aging at a biological, molecular, genetic level, then we have the potential to actually intervene in that process, slow down the molecular changes that are aging, and thereby delay the onset and progression of all of these different declines in function and diseases simultaneously. So that's kind of the big idea. And I like the term 21st century medicine, because I think this is where we're going. I think this is the future of medicine and biomedical research. Instead of focusing on one disease at a time, really trying to keep people healthier longer by targeting the underlying aging process itself. So there's actually been some analysis done to really try to ask the question, well, what might this mean? Let's just say that we can do this in people. And so demographers have asked the question, what would the effect be on life expectancy for a typical 50-year-old woman taking the old approach, one disease at a time, trying to cure those diseases, versus what I call 21st century medicine, which is targeting aging directly. So just based on risk of death from individual diseases, if we consider a typical 50-year-old woman, and if I had, let's just say that what's in this bottle could cure all forms of cancer, and I could give it to you. You can ask the question then, what would happen to life expectancy for a typical 50-year-old woman? It turns out that on average, you only get about 2 to 3 additional years of life expectancy from curing cancer, again, for a typical 50-year-old woman. And I don't know about most of you, but when I first saw that number, I was kind of surprised. I expected that the impact would would be larger. But on a population level, it's actually a relatively modest increase in life expectancy. Even if we could cure 4 of the major killers, cancer, heart disease, stroke, and kidney-related disease at the same time, you only get about a decade of increased life expectancy. So why is that? And it's because if you think back to that circle with all the different diseases of aging, even if we fix 1 thing or 2 things or 3 things, your risk of developing these other diseases of aging continues to go up exponentially as you get older. So if we compare that to the effect now of slowing aging, and this is just based on what's been done in laboratory animals. So if we take what is now fairly routine in mice, for example, in terms of slowing down aging, increasing lifespan, and we just extrapolate that to people, which is a big if. Nobody's proven that we can do that yet. But if we do that, we can take that same 50-year-old woman and we can ask, what would the effect be? And it turns out that you get about 15 to 25 additional years of life expectancy, with the added benefit that those are healthy years, because we've delayed the onset and progression of all of these declines in function and diseases at the same time. And so this is a term that we refer to as health span. So to differentiate from lifespan, lifespan is how long you live. Health span is the length of life that's spent in good health, free from the diseases and disabilities that go along with aging. So we're increasing both lifespan and health span by targeting aging directly. And J.L. Shansky is a demographer who has coined the term the longevity dividend to kind of refer to this idea, this added value that you get from targeting the biology of aging directly as opposed to 1 disease at a time. So again, just to emphasize, the real goal of our research is not so much to increase lifespan. Increasing length of life is great. I mean, I think most people would agree that you don't want to die. You'd like to live longer. But we really want to live longer in good health. And so the goal here is to maximize that period of life that's spent in good health, free from disease and disability. So that's the goal of the research that we're working on. And I just want to show this because I think it makes the point that the scientific community, the research community, still hasn't really caught up to this idea. So what's shown here is fiscal year 2016 funding at NIH. So NIH, National Institute of Health, is where most of the federal research funding comes from, for biomedical research at least. So most of the NIH funding is still focused on individual diseases. So we can consider cancer. This is millions of dollars now for different diseases: cancer, heart, lung, and blood, kidney diabetes, neurodegeneration and stroke, eye disease, dental health, Alzheimer's disease. So one thing probably most of you in this audience don't realize is there is actually an institute at NIH. So NIH has several institutes underneath it. It's kind of an umbrella. There is actually an institute at NIH for aging called the National Institute on Aging, or NIA. It turns out, though, that most of the funding from NIA actually goes to Alzheimer's disease, so to 1 disease of aging. More than 50% of the NIA budget goes to Alzheimer's disease. About another 30 or 40% goes to geriatrics, so clinical geriatrics and behavioral and social studies of aging. And only a very small amount of money goes to the biology of aging, which is really the research to understand what are the fundamental mechanisms of aging. I think this is changing. I think that the scientific community is starting to understand the potential value of targeting aging directly. But still, as of last year, it's a relatively small piece of the pie in terms of research spending that goes towards studying the underlying biology of aging. Now, I know there are other scientists in the room. You may not agree that more money needs to go to aging. I think one thing we can all agree on, though, is that relative to what gets spent on the military, research funding is not that large of a piece of the pie. And our current commander-in-chief would like to see this ratio get skewed even more. So. And I should say, I don't blame Donald Trump. That actually was 2016. That skew has been going on for a long time, but I would like to see it not get skewed any further. Okay, so again, big picture. The goal of our research is to target aging directly, understand what's happening here so we can slow the onset and progression of all of these diseases and enhance health span at the same time that we're also increasing lifespan. So the first point I want to make, the first sort of take-home message I want to make, is that aging is just biology. So evolution has already figured out how to do this, right? So we all recognize that different animals have dramatically different lifespans. And so this is just a smattering of the different animal species that are out there. So you can think about a fruit fly, which lives about 3 months, a mouse, which at least in the laboratory will live 3 years. In the wild, most mice don't make it more than 1 season because of predation. A large dog, which might live 8 to 10 years. A hummingbird, which can live 14 years. This naked mole rat, some of you may have heard of this interesting animal. They live about 30 years. Whales can live 200 years, and these clams that a scientist named Steve Ostad at UAB has been studying can live more than 500 years. So there's dramatic differences in the lifespan of different animals, and people are somewhere in the middle. We're towards the upper end, but we're by far not the longest lived animals out there. So again, nature, evolution has already figured out how to change the rate at which organisms age over many, many orders of magnitude. So this is just a biological problem of understanding, well, what are the genetic factors or environmental factors that cause these dramatic differences in lifespan or aging rates? And even from a very young age, probably everyone in this room, when you were a child, learned that 7 dog years to 1 human year, right? This is something that we just, we accept that dogs age faster than people do. So again, why is that? It's just biology. It's just genetics in dogs that cause them to age faster than people do. So this is a solvable problem. That's the point that I'm trying to make, is that aging is really biology. There's nothing magic about the rate at which animals age. We just need to understand the biology in order to be able to modify it. So the second take-home message that I hope to leave you with is we're starting to understand the biology of aging. So this figure, you don't worry too much about what these things are. This figure is from a review called The Hallmarks of Aging that was published in the journal Cell, which is one of the major scientific journals. And it kind of laid out, in a general sense, many of the advances in the field of aging research over the past couple of decades. And one of the important things that this article did was it sort of classified what we've learned about a handful of molecular processes that really drive aging. Okay, so these hallmarks of aging are conserved molecular processes that are shared across all of those different animals that I showed you that contribute to the declines in function that happen at a cellular level, a tissue level, a whole animal level during aging. Some of these you may have heard of. So telomeres get talked about. There was a recent book that was on the bestseller list by Elizabeth Blackburn about how telomeres contribute to aging. You can actually get your telomeres measured now in a blood test if you want to. I'm not sure that it's particularly useful information, but you can do that if you want to. But all of these molecular processes are hallmarks of aging. They contribute to aging. And what we now know, at least in animal models, is that if we do things to tweak these molecular processes, to speed them up or slow them down, we can change the rate at which laboratory animals age. In many cases, slowing it down to the point where we can extend lifespan by 30%, 40%, even 50% in some cases. So it's possible to slow down the aging process by understanding aging, and we're starting to get there. By no means is this anywhere near being understood at a detailed level, but we're getting to the point where we know enough to actually start manipulating aging in a way that increases lifespan and improves health span. So these are just a list of some of the different ways that scientists have targeted aging in laboratory models. So there are a handful of genetic pathways that appear to be particularly important for modulating aging. And again, what these are really isn't that important for this talk today. The important point is that there are multiple different targets that scientists have shown we can modify genetically to slow down aging and increase lifespan and enhance health span in laboratory animals. There's also several environmental factors or lifestyle factors or pharmacological drugs that we can use to delay aging in laboratory animals. One that you may have heard of is dietary restriction. This is sort of the best studied intervention for slowing aging. And it really just refers to what it sounds like, eating less. So reducing the amount of nutrients that are available to an animal seems to slow down the aging process, at least to a certain point. Obviously, once starvation sets in, that's not compatible with a longer lifespan. But up to that point, you can slow aging by restricting nutrient availability. And we're starting to understand, again, the molecular mechanisms that are downstream of dietary restriction. They're mediating these effects of dietary restriction. And you can actually target those molecular pathways with drugs like rapamycin, which I'm going to talk more about. Exercise is another one that we all sort of recognize, even from, from what we know in people, delays many aspects of aging. And we're understanding at a molecular level which hallmarks of aging exercise is really impinging on. So this drug here is one I'm going to spend a lot of time talking about, rapamycin. It turns out this is actually a drug that targets one of these genetic factors, mTOR, and I'll talk more about that in a minute. Metformin is another interesting drug. This is actually the most widely prescribed anti-diabetes drug in the United States. It turns out that clearly, diabetics taking metformin live longer and are healthier than diabetics taking other diabetes drugs. And there's some evidence that diabetics taking metformin actually live longer and are healthier than non-diabetics not taking metformin, which is consistent with the idea that metformin might actually be, at least to a small extent, slowing down the aging process in people, which fits very well with what has already been seen in laboratory animals. And then a couple of other drugs, NAD precursors. Some of you may have heard of these. You can actually, I'm not suggesting that you go out and buy NAD precursors, but you can buy them on Amazon if you really want to. NA-carbose, which is another anti-diabetes drug. So there are multiple interventions in laboratory animals that have been shown to target the hallmarks of aging and slow the aging process. So I'm going to talk a bit about rapamycin now throughout the rest of my talk. And I'm going to do that because this is clearly the most effective pharmacological method for slowing aging. So it's the most effective drug that we know of to date to slow aging in laboratory animals. And it's a really interesting drug. So this molecule, rapamycin, was actually found on Easter Island, which also goes by the name Rapa Nui. That's where rapamycin gets its name from. It was found in soil samples there. It's produced by a bacterium called Streptomyces hygroscopicus. Rapamycin was originally studied for a variety of different effects. It has been tested widely for anti-cancer effects. It was first approved by the FDA because of its effects on the immune system. So in people, rapamycin is primarily used to prevent organ transplant rejection. Now, it's never used by itself. So rapamycin, you may hear referred to as an immunosuppressant. But it's not really a true immunosuppressant. It's not clearly a true immunosuppressant when given by itself. It's always given with other stronger immune suppressants. What it does do clearly is modulate the immune system in a way that causes your immune system to be less likely to reject a foreign organ. So that's mostly what it's used for clinically. It's also used for some forms of cancer as well. So we've been interested in rapamycin for a long time because what we've found and what several other labs have found is that if you give this drug at lower doses to organisms in the laboratory, you can actually increase their lifespan. And you seem to delay these hallmarks of aging. In fact, rapamycin's interesting because it actually hits all of those hallmarks of aging that I showed you before on that list. So it seems to broadly delay these molecular processes of aging, at least in laboratory animals. And this is true in a single-celled organism, budding yeast, in these nematode worms called C. elegans, Caenorhabditis elegans, in fruit flies, and in mice, that treating these animals with rapamycin makes them live longer and seems to slow down the aging process. And in mice, you can actually look at measures of health span in a fairly comprehensive way. So mice get many of the same diseases of aging that people do. And so we can ask, what is the effect of rapamycin on health span in mice in a way that's harder to do in a worm or in a fruit fly? And it turns out that in every health span metric where rapamycin has been measured, there's evidence that it can slow down the aging process. This is true for cognitive function, so mice lose brain function with age, much like people do. Rapamycin seems to delay that. It improves muscle function. I'll talk a little bit about heart function, a little bit more. It improves heart function. I'll talk a little bit about immune function. It improves immune function during aging, improves stem cells. A variety of different health span measures in mice are improved by rapamycin. Interestingly, it also seems to delay the onset and progression of Alzheimer's disease in multiple mouse models. And I'm just going to take a very brief aside because I want to hit this point a little bit harder. This is a source of particular frustration for me in that in every preclinical model of Alzheimer's disease, so by preclinical I mean an animal model, in every preclinical model of Alzheimer's disease in mice, rapamycin has been shown to be beneficial. And in many cases, it's beneficial after the symptoms of the disease set in. So it's quite striking, actually. I don't know of any other intervention in mice that has this track record in preclinical studies. So you might think, given that this is already FDA approved, somebody must be testing this in a clinical trial, right, for Alzheimer's disease. So this is a result from the clinicaltrials.gov. This is the database of clinical trials. So you probably can't see this, but there are 1,993 studies for rapamycin, 1,969 studies for Alzheimer's, 0 studies for rapamycin and Alzheimer's disease. I do kind of understand, and if anybody's interested, I'll be happy to talk to you about it. Let me just say it's very frustrating to me that rapamycin is not being tested in a clinical trial for Alzheimer's disease. So I'm pushing to try to get that to happen, but there are lots of reasons. Part of it has to do with the fact that rapamycin is off patent, so there isn't a lot of money to be made for this, but it's frustrating that this is not being done. And some people will say that the mouse models of Alzheimer's disease aren't very good, which is true. My response to that is, why are we using mouse models for Alzheimer's disease if you're not going to actually be guided by them to test things? So in any case, hopefully that will happen, because I think there's a pretty good chance. Obviously, we never know. But given what I know about rapamycin and its effects on mTOR and the biology of how rapamycin is working, I think there's a pretty good chance that it could be effective, at least in people who are in the process of developing full-blown Alzheimer's disease. So back to aging, away from Alzheimer's disease and back to the biology of aging. One of the really exciting things about rapamycin is in mice, it was shown back in 2009 that you could even start treating a mouse in middle age with rapamycin and still increase its lifespan. And this was really the first time that any drug had been demonstrated to increase lifespan in a mammal when you start it in middle age. So in this particular experiment, so first of all, I'm not going to show a lot of data today, but let me take you through what's shown here. So these are typical what we call lifespan curves or survival curves. We use them a lot to look at data in studies of aging. And what's shown here is the fraction of the population that's alive, so survival, as a function of age. So if you look out here at 600 days of age, 20 months, that's about a 60-year-old mouse. And we go up here, about 90% of the animals are still alive at that age point, which is roughly the same in people, right? If you think about the population as a whole, at 60 years of age, about 90% of people are still alive. So what was done in this experiment was they started treating the rapamycin-treated mice, which is this line here, the red line, at 20 months of age, so about the equivalent of a 60-year-old person. And then the controls just stayed on their same diet. And you can see the controls die off by about 900 days of age. Half of them are no longer alive, whereas many more of the rapamycin-treated mice are alive. So their average lifespan is about 1,100 days of age. So it increased average lifespan quite significantly, even when we started at the mouse equivalent of a 60-year-old mouse. So this was done by the National Institute on Aging Interventions Testing Program, which is done at 3 different sites around the United States at the same time. So it's got threefold replication built into the design of the study, which is really nice because it tells you that this is a real result. All 3 sites saw the same thing. You get about the same effect when you start with young mice. So this is now starting rapamycin at 9 months of age. And you can see the difference between the dark line and the red line is about the same here as it is here. So the mice lived longer whether we started rapamycin treatment at 9 months of age or 20 months of age. And it's by about the same amount. So you get most of the benefit at least for lifespan, from rapamycin treatment, even when you start in middle age. And if you're thinking about an intervention that we might someday want to use to delay aging in people or in dogs, it's very appealing if we don't have to start treating the people or the dogs with the intervention early on, right? Because your risk of developing side effects is much greater if teenagers have to start taking this drug. But if you can wait till you're 60 or 65 to start taking the drug, your risk of side effects goes down, and if you can still get the benefits, that's great. So this is kind of appealing from a translational perspective, the idea of actually moving this into a clinical setting. Okay, so why aren't we all taking rapamycin? It delays aging, makes you live longer, makes you healthier. It has side effects, right? Any drug is going to have side effects. And in people taking rapamycin to prevent organ transplant rejection, there is a long list of side effects. This is not all of them. If you go on Google, you can see the whole long list of side effects. None of them are really life-threatening, but they're not pleasant. So what's called the dose-limiting toxicity, why most patients stop taking rapamycin, is because they get sores on the inside of their mouth, and that's not a fun thing to have, right? So, so there are side effects. I think it's important to consider that the data we have for side effects with rapamycin, though, are sick people. There's a reason why they had an organ transplant, and they're taking high doses of the drug. What we really don't know is that at lower doses of rapamycin, would these side effects still be there? And if so, would they be at a much reduced level that would not be incompatible with taking rapamycin to improve health span or extend lifespan? There's a little bit of data from one study that's been published that suggests that the side effects are probably much lower in healthy people taking lower doses. But at this point, we really don't know. So this led us to think about the idea that, well, maybe we could get, so we know that we can start rapamycin late in life, at least if you're a mouse, and get the benefits. Maybe we could only give it to the mice for a short period of time. So do a transient treatment of about 3 months and get some of the benefits for lifespan and health span. And there was actually some data in the literature already supporting that. So there was one study in mice showing that 3 months of treatment with rapamycin can improve heart function. So just like in people, if you take a mouse, the function of their heart goes down with age. If you give them 6 weeks of treatment or 10 weeks of treatment with rapamycin, you can actually restore those measures of heart function. And again, the data here aren't all that important. You just have to take my word for it, that for at least these measures of heart function, which is done by a method called echocardiography, it's like an ultrasound for the heart, short-term treatment with rapamycin can cause a young mouse heart to function more like an old mouse heart. The same thing seems to be true for immune function, and I will take you through the data here because I think it's a really interesting experiment. So this was done by Pan Zhang, who's at Michigan, and so again, just like in people, the immune function of a mouse declines with age. So if you give an old mouse a vaccine, they're not going to respond to it as well as a young mouse does. Exact same thing is true in people, okay? So what they did in this experiment was they asked, well, what's the effect for a flu vaccine if we give the mice 6 weeks of rapamycin before we give them the vaccine? So the design here is pretty simple. They take an old mouse, I think in this case it was 24 months old, give them 6 weeks of rapamycin, then they wait a couple of weeks, then they give them a flu vaccine, then they wait a couple of weeks, and then they give them the flu. And they ask, did the vaccine work? Okay, so that's the logic of this study. And this is the results. And again, this is a survival curve. So this is the percent of animals that are alive as a function of age. In this case, it's days. So if the animals got the vaccine and they were protected, they'll survive the challenge with the flu, getting influenza. So if you're a young mouse and you don't get immunized, that's this solid line here. You get the flu, you're not protected, you die. If you're a young mouse and you were immunized, you respond to the immunization, you're completely protected when you're challenged with the flu. So all of those mice survived because they got the immunization. If you're an old mouse and you don't get rapamycin, but you do get the immunization, that's this dotted line here, most of them die. So they got the flu shot, they got the immunization, but because they're old, their immune system did not respond to that immunization. Most of them were not protected, and they suffered when they were actually given influenza. If you're an old mouse and you got rapamycin and the immunization, that's a dotted line you can't really see here. It's buried under this solid black line. They were all protected. So just that 6 weeks of getting rapamycin in an old mouse was enough to cause their immune system to respond to the vaccine and be protected. You could imagine why that might be very valuable in elderly people who are recommended to get a flu vaccine, even though a lot of the time it doesn't work because the elderly immune system doesn't respond very well to the vaccine. So Novartis has actually published a study showing that the same thing seems to be true in people. That's this paper here. So this was the one study I mentioned where they looked at healthy elderly people for 6 weeks, and they saw a similar improvement in response to flu vaccine. Now, these were people. They did not give them influenza to test it. They looked at antibody responses to the vaccine, but it seems to work the same way. So that's encouraging that rapamycin might actually have similar effects on immune function in people as it does in mice. Okay, so we did an experiment where we really just looked at aging. So we took 20-month-old mice. We gave them 3 months of rapamycin. So again, remember, this is about the mouse equivalent of a 60-year-old. We gave them 3 months of rapamycin or a control, and then we didn't do anything. Then we just let them age out the rest of their life. So really the only difference in these animals was they either got a control for 3 months, 20 to 23 months, 60 to 65 years old, or they got rapamycin. And then we looked at lifespan. And so again, survival, how long they lived, age. The mice that got rapamycin had about a 60% increase in life expectancy after the end of the treatment. So even just this 3-month period of treatment with rapamycin was enough to have a pretty dramatic effect on life expectancy following the treatment. And in terms of absolute length of time, that's about 141 days in this particular cohort, which is interesting, I think, that the lifespan extension following the treatment was actually bigger than the length of time they got the drug. What does that mean? If we just make sort of a simple argument that rapamycin stops the aging process, just halts it for the length of time you're getting it, we would expect the extension of lifespan to be equal to the length of time that the animals got the drug. It's actually bigger, which suggests that at least to some extent, there's a rejuvenation process, at least for certain tissues and organs, that accompanies the effects of rapamycin. And we kind of understand, we're starting to understand why that is. So obviously we have no idea if the same thing is going to be true outside of the laboratory in other organisms, but if it is, that might mean about 20 years for a typical 50-year-old woman, or it might mean 3 to 4 years for a 7-year-old dog. So that's pretty substantial if rapamycin has anywhere near the same effects on life expectancy in either dogs or people. And so that's what brings me to sort of the last part of my talk, which is how we're now starting to extend some of what we're learning in the laboratory into the clinical setting. And this was really motivated by this bigger question, which a lot of people in the field are thinking about, which is how do we do that? How do we get from what we've learned in the laboratory to actually being able to make a difference in people's lives, to bring some of this into the clinic? And that's a really hard problem when we're talking about aging for a variety of reasons. The approach that most people are thinking about are clinical trials, which is the logical approach in humans. But there are some real challenges with that. One of them is that people live a long time. That's a really long clinical trial if you want to look at aging as your endpoint, right? Especially if you want to look at lifespan. So the other approach that people are thinking about is to try these interventions in people who have an age-related disease already. Because there, you can, it's sort of a more traditional clinical trial model. You take sick people, you treat them, and you see if their disease gets better. That might work. I've already told you that I think rapamycin might work for Alzheimer's disease. But it might also be the case that once the disease has gotten to the point where it's clinically diagnosed, that's not necessarily normal aging anymore. And it's not really clear that the same hallmarks of aging are going to be impacting the progression of a cancer or diabetes or other diseases once it's gotten to the point of clinical diagnosis. So there's reasons to be concerned that that approach might not work. So what we would really want to do is start in populations that are healthy. They're older, so they're at risk for these diseases, but they're healthy, and ask, does an intervention actually delay the aging process? That's hard for a variety of reasons. So our approach is really to start with our pets, with companion dogs. And so this is the Dog Aging Project. If you're interested, this is the website. This is one of my dogs. His name is Dobby. I have 3 dogs. He's actually my favorite. I'm not really supposed to say that, but I don't have favorites with my boys. I have 2 boys, and I love them both equally, but Dobby is my favorite dog. So the idea is, can we start to take some of what we've learned about aging in the laboratory and apply it in pet dogs. And so there are really two goals to the dog aging project. The first goal has nothing to do with interventions, has nothing to do with rapamycin. The goal here is really to study aging in dogs and try to understand at a very detailed level what are the genetic and environmental factors that influence healthy aging in dogs. What are these factors? Like I talked about before, just like in people, different dogs age differently, even if they're the same breed, right? What are the genetic and environmental factors that cause some dogs to age well, live a long time, stay disease-free, and other dogs not to? And I'll tell you how we're approaching that. And then the second part, which I'll spend a little bit more time on, is to really ask, can we slow aging in dogs? And using rapamycin as our first approach to ask, does rapamycin delay aging in pet dogs to increase healthy longevity? So this is the dog aging project team at this point. So Kate Creevy is our lead veterinarian. Daniel Promislo is a professor in pathology and biology. He's the co-director of the dog aging project. Sylvan Erfer is sitting in the front row here. He's a postdoctoral scientist working on the project. This is my wife, Tammy. Dobby is also her favorite dog. This is Kelly Jin, who's a graduate student in Daniel's lab. And so this project so far has been supported by a grant from the Donner Foundation, by the Irish Wolfhound Association of New England, a little bit of money from NIH, and many fantastic donors who have contributed to this project. So first of all, why? Why do we think it's a good idea to study aging in dogs? And I think there are a lot of reasons why this makes a ton of sense, right? So first of all, again, how many of you have dogs or have had dogs in the past? So you probably all recognize that dogs age a lot like we do. They get many of the same functional declines that people do, many of the same diseases of aging that people do. It's not identical. They don't get the diseases of aging that we do in the same percentage necessarily that we do, but they do get all of the same diseases of aging that we do, and their risk for those diseases goes up with age. But they do it faster. So that means that with something like rapamycin, instead of having to wait 20, 25 years to know the answer, we might be able to know the answer in 3 to 5 years, which for those of you who know something about NIH funding cycles, 5 years is kind of the magic number, because that's how long an NIH grant typically lasts. So there's a grantsmanship component to this too. So we can know the answer in a reasonable amount of time. Dogs have this fantastic genetic and phenotypic, trait diversity. So we know a lot about the genetics of dogs. We know about the purebred structure that dogs have. And then we've got this mixed breed population, which is really cool and genetically diverse. And in terms of traits, I guarantee you everyone in this room can tell the difference between a Chihuahua and a Great Dane, right? I mean, there's huge variation for almost any trait you think about. So that's really powerful because it allows us, again, to map genes and environmental factors that influence these traits. And the same thing's true for lifespan. A Chihuahua is going to live, on average, about twice as long as a Great Dane. So there is variation in lifespan, just like there is many other traits. Probably the most important one, though, that we can't capture in basic science studies is dogs share our environment. And for those of you who raised your hands, you don't have to raise your hands again, but I would bet several of you have allowed your dog to sleep in your bed at one point. Right? It's hard to imagine an animal that would share our environment to a greater extent than our pet dogs. And that's really important if we're also interested in understanding aging in humans. People love dogs. The people who participate in our study are true citizen scientists. They help us gather data. And I think if we're able to slow aging in dogs, this will help convince people that this is not science fiction. This is science fact. This is reality, that it is possible to slow the aging process and probably possible to do that in humans as well. And then I'm a pet owner. I'm a dog lover. I love my dogs. If we can improve the healthy longevity of pet dogs, that has a direct impact on the quality of life for pet owners by giving them more quality time with their pets. So I believe that there is a sort of intrinsic value to doing this as well. Okay, so the longitudinal study of aging is the approach to really map the genetic and environmental factors that influence healthy aging in dogs. The goal here is to enroll more than 10,000 dogs nationwide and follow them throughout their life to understand what is it that causes some dogs to age well and some dogs not to age well. We plan to capture genetic variation, so this will be open to all dogs in principle. So purebred dogs, mixed breed dogs, we want to get a diversity of genetic backgrounds, again, to really be able to understand what are the genes that influence healthy longevity in dogs. We also want to capture environmental diversity. So we want to get rich dogs, poor dogs, healthy dogs, dogs with questionable lifestyle choices, and really understand, again, what are these environmental factors that influence aging in dogs? So for example, we'll be able to monitor the activity of the dogs because they'll have a GPS tracker in their collar, and we'll see how much exercise they're getting, things like that. We'll know where they live. We'll get owner survey information. We'll get water quality and air quality from the location that they live in. Most people don't give their dog bottled water. They drink water from the tap. But if they do give them bottled water, we'll get that information from the surveys. So this will be a very powerful and rich data set. And I don't want to take you through all the different things that we're going to do, other than to say this is going to generate a boatload of data. There's going to be a lot of data generated from this project. And our goal is really not just for us to be able to look at that data, but to make it available to the broader community, to really make this an open science project, and to think of clever ways that we can put this information out there on the internet so that other scientists may be interested in aging, may be interested in something else, can actually get that data and use it and make discoveries from that data. So this is not only about us, but it's about really making this available to the wider community. Okay, so if you're interested, you can go to the website and nominate your dog to participate in either the longitudinal study or the rapamycin trial. Again, it's dogagingproject.com. I forgot to mention it. If anybody wants, there's some cards in this box up here. You're welcome to take one. That's got our website and my email on it. We have about 5,000 people or so who have already nominated their dogs, so we want to get to 10,000, so there's still room if you're interested. Okay, so now I'll just finish up in the next few minutes and tell you what we've done so far with rapamycin. So this is now the other part of the dog aging project. This is really asking, does rapamycin slow aging in pet dogs? So the idea here is, first and foremost, these are people's pets. We want to make sure we don't hurt anybody's dog. So we're starting with low doses of the drug that, based on veterinary colleagues, we're confident are not going to have significant side effects. And we want to ask, if we start with low doses of the drugs in middle-aged dogs, so they have to be at least 6 years old, does rapamycin have any effects for health span, diseases of aging, hopefully lifespan as well? And so that will take, again, as I mentioned, about 3 to 5 years to really answer the question. So we've done one study, which was here in the Seattle area. It was a VCA veterinary clinic in Linwood, where we did a 10-week study. And this time frame was kind of based on what I already told you about in mice with heart function and immune function, and where we knew that over this time period, at least in mice, that was enough to see some beneficial changes. So it was a 10-week study. 40 dogs came into the study. Again, they had to be at least 6 years old. They had to be at least 40 pounds, and they couldn't be sick. So this is a study of healthy aging. So we wanted to start with dogs that were relatively healthy. And these are 3 of the dogs that actually participated in the study. So the design, this is a little bit complicated. The main points I want to make is they come in for an initial exam. If they're found to be healthy, they get randomized. This was a double-blind, placebo-controlled study. They have another exam after 3 weeks just to make sure everything looks good. And then after 10 weeks, they stop the treatment, and they come back in for their last exam. And the main things that we got at these exams were blood chemistry, so sort of the standard blood panel that a veterinarian is going to look at, a geriatric blood panel. And we looked at heart function using this non-invasive echocardiogram method that I mentioned for mice. So we wanted to ask the question, would 10 weeks on this drug have any impact on heart function in dogs, as had previously been seen in mice? So a couple of take-home messages. There were really no changes in blood chemistry, and we didn't find any evidence for an increase in side effects in the dogs that received rapamycin. This is a kind of an ugly table. These are all the different side effects that owners reported. So our owners sent in surveys every week. One of the reasons why I showed this is any of you who've had old dogs will recognize that there's a good chance that in a 10-week period, your old dog is going to have one or more of these side effects. So that was part of the reason why it was important to have a placebo, so we could ask, was there any difference between the treatment groups and the placebo group? The other thing that I'll mention, we don't know if this is real, but among the positive traits that or effects that owners reported, more of the rapamycin-treated dogs, their owners reported increased energy. That could be interesting. And the one that I really was a little bit was unexpected, I was surprised by, was 40% of the lowest dose rapamycin and 20% of the higher dose of rapamycin, the owners reported their dogs were more affectionate. I don't know. None of the owners in the placebo group reported that. So it's interesting. It might actually suggest effects on cognitive function, but we really don't know at this point. So these are the data for heart function, okay? So this is, again, I know it's a complicated slide. What's shown here is before and after. So anything on this dotted line means that that dog did not change over the 10-week study. Okay, anything below the dotted line, function got worse. Anything above the dotted line, function got better. The blue are placebo, the red are the rapamycin-treated dogs. What hopefully you can appreciate is more of the red triangles are above the line, more of the blue triangles are below the line. And then this is just a different way of showing the data. So just like in mice, it looks like rapamycin is improving this measure of heart function after 10 weeks in old dogs. And we saw the same thing for 2 other measures of heart function. So it looks consistent with the idea that rapamycin is improving heart function in dogs. Now, I'll just mention, any of you who are really into data, this study is published. It was published in the journal Geroscience, so you can go online. It's open access. You can get the paper and actually look closely at the data if you want to. So phase 2, I really hope, is starting next month. So this is a 1-year study that will be done at Texas A&M University. We've had the funding for this study for more than 9 months now, and it's really just been the institutions talking to each other that's held this up. A little bit of frustration, but we're almost there. So hopefully that will start next month. This will, again, be a 1-year study, 6-month treatment, 6-month follow-up. This is a longer treatment. Again, we'll look at heart function, but we really want to start to look more broadly at aging. So we're also going to look at cognitive function. We're going to look at periodontal disease and activity with those GPS trackers. And again, this was funded by the Donner Foundation. This is my second dog. Her name is Chloe. And then this is my third dog. Her name is Betty. She's a new addition to our family. She was a rescue that we got about a year ago. So where we want to get to is phase 3. This is the 5-year study. This is the study to really answer the question, does rapamycin slow aging in dogs? Does it delay multiple declines in function and diseases that go along with aging, and does it extend lifespan? So hopefully we'll get there. We have an NIH grant that's being reviewed next month. If that gets funded, then we'll be actively enrolling for this study. And again, you're all welcome to nominate your dogs to participate if you would like to. So with that, I'll finish up with my last take-home message. This is also my last slide. And I just want to make the point that, for me personally, given everything I know about aging and biology, I absolutely believe that this is doable. And I actually don't think it's going to be that hard to slow aging in dogs. I don't know if rapamycin is going to have the same effects in dogs that it does in mice. But I showed you that slide of multiple different ways that scientists have found to target the biology of aging in laboratory animals. Everything I know about biology makes me believe the same thing is going to be possible in dogs. And it's really just a matter of putting the resources and the effort towards that problem. And I think it's a problem that's worth solving. So I think we'll get there. I don't know how long it'll take, but I think this is a solvable problem. And I think that we have the chance really to be able to increase healthy longevity in dogs by 3 to 4 years in the relatively near future if the sufficient resources are put towards that. And I show this picture because this is a picture that was published in it's a web journal, I think, called Fusion, where they somehow got this picture of me off the internet, which I did not give them permission to use, with my dogs, and they photoshopped halos around the dogs' heads. And the title of this was, "This Scientist Wants Your Dog to Live Forever," which is a very unfortunate title. I mean, it's true. I would love for your dog to live forever. I would love for my dog to live forever. That's not what we're talking about. We're talking about very realistic and feasible changes in the biology of aging that I think still 3 to 4 years, 10, 15%, that's a pretty significant impact in dogs. And ultimately, it will be a significant impact in people as well. So with that, I'll stop. Happy to take questions, and I'll stick around for a while afterwards if anybody wants to chat. Yeah.